Welcome everyone to this journal club. This is one of the journal club sessions of our Memory Decoding Challenge series, part of the Aspirational Neuroscience Prize. And today we're going to be discussing the paper Reconstructing a New Hippocampal Engram for Systems Reconsolidation and Remote Memory Updating by Li et al. And as mentioned, it's pretty dense, so I hope you don't mind if I might seem to be speaking a little bit quickly, but please slow me down if things are getting out of hand. All right, so this study attempts to elucidate the mechanisms that are underlying systems reconsolidation, so the process by which remote neocortex-dependent memories are re-engaged by the hippocampus upon recall. And the authors did this by developing a novel triple event labeling tool and employing two-photon imaging. And with that, they demonstrate that remote memory recall recruits a new hippocampal engram ensemble rather than reactivating an old one. They were able to study this process for both re-engagement with consolidated episodic memory, as well as the process of updating such memory with new context information. Both situations where they showed that hippocampus was re-engaged in a manner that's similar to its engagement during initial learning, but using different populations of hippocampal neurons. This recruitment is facilitated by adult neurogenesis mediated silencing of the initial engram, allowing the integration of new contextual information into the existing memory. And furthermore, the reconstructed hippocampal engram coordinates with the amygdala via medial prefrontal projections that enable memory updating. And these findings reveal that system reconsolidation involves dynamic engram reconstruction, providing a neural basis for how remote memories are adaptively modified with new experiences. And I think that this tracing they did from acquisition through reconsolidation seems like a candidate for at least an exploration of a memory decoding process. Now, before diving right into their paper and their figures, I thought it might be useful to glance briefly at a mouse brain and at the hippocampus. You can see it here in green in the mouse brain. It's more accessible in the mouse brain than in a human brain. And to also have some idea, for instance, of the layout of the hippocampus, of the different regions, like CA3 and CA1, and the dentate gyrus, this little blue thing shown here in another diagram. You've got, see again, this sort of little coiled situation you've got going. I'm going to discuss mostly this image on the right, the one that has the least pretty colors. It's a model from the Hasselmo-Eichenbaum lab, and I just wanted to point out a few things in it. So memory consolidation or memory reconsolidation, including recruitment from neurogenesis, fits into existing models of hippocampus and medial temporal lobe episodic memory function. And the figure here shows the function of different hippocampal subregions as presented in models, for example, from the Hasselmo and Eichenbaum labs. And this one here, A, is a summary of the anatomical structure of the hippocampus. The input from and output to neocortical regions is through the entorhinal cortex. Sorry, I've got a little issue I've got to clear up here. Don't know what's going on there. Oh, sorry. What's happening? Oh, okay, great. Everything's normal. Yeah, so input from and output to neocortical regions is through the entorhinal cortex, with major input from layers 2 and 3. And the entorhinal cortex projects to dentate gyrus and to regions CA3 and CA1 via what's called the perforant pathway. The dentate gyrus projects to region CA3 through so-called mossy fibers. And CA3 is that region that has the extensive recurrent excitatory connections and projects to CA1 via so-called Schaefer collaterals. The region CA1 then projects back to entorhinal cortex directly and also through the subiculum to layer 5 of entorhinal cortex. So everything's going in and out through entorhinal cortex there. And B is a summary of the function of different subregions within the hippocampal model. It's a bit dense and I won't get into all the details, but so the dominant anatomical features of the hippocampus are consistent with the notion that inputs from entorhinal cortex are processed at successive levels from the entorhinal cortex layer 2 spiny stellate cells to dentate gyrus granule cells two recurrently connected CA3 pyramidal neurons onto CA1 and subicular pyramidal cells, along with a direct projection from entorhinal layer 3 and fed back to entorhinal layer 5. And then the medial prefrontal cortex feeds to entorhinal cortex, CA1, and then subiculum feeds back to prefrontal cortex. So we've got this other big circuit, this other loop going on there as well. All right, with that as a context of where all this stuff is taking place, and I see there's already a hand up. Whose hand is that? Yes, Ken.
Yeah. So maybe you just said this, but connection to the amygdala, I think, would also be.
That would have been useful to have in there as well. Yes, given that that's what they get into as well. Yeah, yeah.
Oh, any idea, is that through entorhinal cortex or?
No, I don't believe so. No, I think that's a separate, if I'm not mistaken. But again, don't quote me on this.
Okay, thanks.
Just don't quote me on that, because I mean, obviously it's being recorded, but try not to quote me. All right, let's dive in. Figure 1, or Figure 1A, is illustrating the behavioral paradigm that was used to study hippocampal re-engagement during remote memory recall. First, the mice underwent conditional fear conditioning, contextual fear conditioning, forming an initial memory. Then 21 days later, at a time point when memories are typically systems consolidated and neocortex dependent, they were re-exposed to the context to trigger remote memory recall. This recall reactivates the medial prefrontal cortex engrams, and that was shown in other work. Finally, 1 day after recall, memory was tested in either the original context, called OCTX, or a generalized context, GCTX, to assess retrieval specificity. And this design allowed the authors to investigate how remote memory recall re-engages the hippocampus and whether new information is incorporated during reconsolidation. So figures 1B to E are shown here. These demonstrate the necessity of hippocampal activity in remote memory reconsolidation. First, in figure 1B here, they targeted the dorsal dentate gyrus with a viral vector AAV, CAM-K2A, and then blah, 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 more stuff, to optogenetically silence hippocampal activity. In figure 1C, by suppressing dentate gyrus after remote recall during the test phase, this impaired memory retrieval, confirming that the hippocampus is re-engaged post-recall. And in figure 1D, it shows that when they blocked dentate activity during remote recall itself, subsequent test phase inhibition had no effect, meaning hippocampal re-engagement was prevented. And then in figure 1E, it reveals that blocking hippocampal activity during recall also prevented memory degeneralization, so the behavior is not context specific. Normally, remote recall restores contextual specificity, so that's a hippocampal dependent feature. But without hippocampal involvement, memories remain generalized, consistent with neocortical dominance. And together, those findings showed that hippocampal re-engagement during remote recall is critical for both memory retrieval and updating contextual specificity. Heading into figures 1F to 1J, this explores whether blocking hippocampal re-engagement during remote recall shifts memory dependence back to neocortex. In figure 1F, they co-targeted the dorsal dentate gyrus and the medial prefrontal cortex with that viral vector, AAV something something, to independently inhibit each region. Figure 1G shows that after normal systems reconsolidation, where the hippocampus is engaged, silencing medial prefrontal cortex during the test phase had no effect on memory retrieval, confirming that memory was hippocampal dependent. However, when they inhibited the hippocampus during remote recall, figure 1H, subsequent medial prefrontal cortex suppression did impair memory, proving the memory had reverted to neocortical dependence. So then in I and J, they reinforce this by demonstrating that the same dichotomy is also true in contextual specificity. So in 1I, normally memory remains intact with PFC inhibition, but with prior hippocampal blockade in 1J, the PFC becomes critical for retrieval. And those results then confirm that hippocampal re-engagement during recall is necessary to shift remote memories back to hippocampal control. Otherwise, they remain neocortex dependent. All right, so figure K, this is outlines the experimental timeline that's used to validate the functional role of the hippocampus in systems reconsolidation through targeted manipulation of new protein synthesis. And the key phases in the protocol are the contextual fear conditioning to establish initial memory, a 21-day consolidation period, allowing the memory to transition to a neocortex-dependent remote state, then remote memory recall on day 21, which triggers hippocampal re-engagement, and precise pharmacological or genetic intervention is done during the critical post-recall window to disrupt the protein synthesis, which is required for memory reconsolidation. So then memory testing happens 24 hours later to assess the functional consequences of blocking hippocampal plasticity. And then this design enabled them to causally link protein synthesis in the hippocampus to reconsolidation and updating of remote memories. Figures 1L to 1Q demonstrate the protein synthesis dependence of hippocampal re-engagement during systems reconsolidation. And in figures L and O, top and bottom, this is where we're seeing the experimental approach, or they're applying inhibition, applying the suppression of protein synthesis in the second case. So the authors used the viral delivered TET-on gene expression system to express a genetically encoded protein synthesis inhibitor in either the dorsal dentate gyrus, that's figure 1L, this case, or in the medial prefrontal cortex, that's case in O here. And this allowed temporally precise inhibition of protein synthesis in each region. And the key findings of that are, so now we're getting into the next bits here, that in figures 1M and 1N, hippocampal, so dentate gyrus, protein synthesis is required. I like to say dentate gyrus when I can because I don't want to lump them all together. CA1, CA3, dentate gyrus do quite different things. But okay, it's dentate gyrus protein synthesis. Blocking protein synthesis in the dentate gyrus impaired memory reconsolidation, confirming that hippocampal re-engagement depends on new protein synthesis. PFC protein synthesis is dispensable. So in P and Q, inhibiting protein synthesis in medial prefrontal cortex had no effect, showing that reconsolidation does not rely on medial prefrontal cortex plasticity. And this optogenetic evidence aligns with prior lesion and pharmacology studies and reinforces that the hippocampus is functionally recruited during remote recall, and that this re-engagement is activity dependent and requires hippocampal protein synthesis. And the reason why this matters is that it clarifies that remote memory updating relies on hippocampal plasticity, not just neocortical circuits, providing mechanistic insight into how old memories are modified. Sorry, in the background, you might be hearing some cat sounds. Hope you can ignore those. And I see, Ken, that you have a question.
Yeah, so there's a lot.
Yeah, it was.
I don't think I'm the only person that could could need a little bit of a summary here. Is it correct to say that essentially the hippocampus is overrides? It's like if the hippocampus recognizes what's going on, it gets control. But if the hippocampus doesn't recognize, then the medial prefrontal, if it recognizes, gets control.
Sorry, it sounds like that to me, but it's not clear to me that they showed a mechanism for that, or that that was even included in this study. So they're just showing that if the hippocampus is not able to recall memory or to use the memory that is already remotely instantiated, so that's already in the neocortex, if it's not able to pull that back and then take control, as you say, then the fact that that memory is there, if medial prefrontal cortex is still working, can be used without the kind of specificity that the hippocampus might be able to deliver, say in a new context, for example. So the way I think of it is that if the hippocampus exists, if it's available, then you can use the mechanisms that were there for the initial learning to do nice, cool things with the memory. You can bring it back and you can update it, modify what it means, by attaching it to another context, that sort of thing. If you don't have the hippocampus, if that's not available, then you still have the old memory, but only with what it can do, what the neocortex can do by itself. But what's not clear is that there's any real discussion of a model of how this switching happens or how the hippocampus dominates and takes over control. So there would be a need for a little bit more explanation there, I think.
Okay, I was just saying from the point of view of summarizing the results. But yeah, I agree. There would have to be a mechanism.
Yeah. Alex.
Alex, you might be muted.
No, Alex typed it in the chat. Alex is saying, did they explain what the protein
I just
synthesis?
posted my question in the chat.
Yeah, what the protein synthesis for what? New synapses or something else, internal cellular components needed for engrams. So typically, protein synthesis is something you need when you're doing learning. So plasticity, LTP, and then eventually new synapses. So that's quite general. But what they're doing is they're specifically targeting the protein synthesis inhibition in particular places. So either in dentate gyrus or in medial prefrontal cortex. So then it's not brain-wide shut off of protein synthesis, but very specific shut off of protein synthesis. And that's how they're pulling out whether or not new learning, plasticity, and eventually synaptic changes are needed for the task that's taking place, the memory task that's going on. And then they're looking at whether it's dentate gyrus, where the plasticity is most needed, or prefrontal cortex. So that's precisely what they're trying to figure out. But if this was to say, oh, it's not just the reactivation, it's not just about the activity in those regions, but it's synaptic plasticity that's required. So really, we can just say, in short, testing protein synthesis is just testing plasticity being involved. All right, I will carry on, and
Okay, thank you.
I'm going to take a sip because clearly I'm losing my voice. Okay. This is still, am I still on that one here? No, I'm actually past it. I went on too far. Okay, we're at figure 2. Oh, 2 out of 7. We're going strong. So this is figures 2A to D. They identify the origin of reinstated hippocampal engrams. And this is where they introduced this new tool they developed, shown, in 2A and 2B. These two pieces are describing what they're doing with their new tool. This is to track memory engrams across 3 events, training, remote recall, and the post-recall test. The authors developed FOSS-TREE, a novel triple event labeling system. They use FOSS-TRAP. TRAP means targeted recombination in active populations to track and trace the label neurons involved in training, so the first event. FOSS-TET-TAG to label neurons involved in remote recall, so event 2. And CFOS immunostaining for the post-recall tests involved cells, detecting that. And this is a system that enables precise sequential tagging of active neuronal ensembles. And yeah, I couldn't quite make heads or tail of B here, but I think this is just their injection protocol. So validation experiments confirmed separately in the supplemental materials that this FOSS-TREE is specific, so the specificity of it, and that it works in various behavioral stimuli. The key findings here are shown in figures 2C and 2D, and they are that remote recall does not reactivate original training engrams. In the dentate gyrus, both remote recall and post-recall tests activated distinct ensembles, not the neurons recruited during initial training. Instead, the post-recall test reactivated neurons from remote recall. And that suggests that new engrams are formed during reconsolidation, replacing the original trace. And you could contrast that a bit with recent memory and prefrontal cortex dynamics. In recent memory recall, that does reactivate training engrams, and they did that in their supplemental materials as well, highlighting a fundamental difference between recent and remote memory mechanisms. And in the medial prefrontal cortex, remote recall reactivated training engrams, so just what you would expect, but the post-recall test did not reactivate either ensemble. Again, that was in their supplemental materials. I'll just quickly finish what I was going to say for this slide, and then I'll take that. So the systems reconsolidation reconstructs hippo engrams, recruiting new neurons to update remote memories. And that aligns with their earlier findings, shown in figure 1, that hippocampal protein synthesis is critical and provides direct evidence of engram turnover. The remote memories are not simply reactivated, but reconstructed in the hippocampus using new engrams. And so the prefrontal cortex and hippocampus play divergent roles, as we've already discussed. And the interpretation that they give it is that the prefrontal cortex stabilizes original traces while hippocampus generates new ones for updating. Personally, from prior work, I feel this fits into the notion that dentate gyrus is highly involved with novel context learning and needs to enable distinguishing novel contexts with potentially overlapping details. And now I will take that from Alex. Yes, so Alex has a question. Oh, hasn't typed it yet.
My question briefly was just, I think that I read this properly, but it's just the dorsal dentate gyrus they were looking at there, right? They weren't seeing if there was new neuron recruitment or not in the prefrontal cortex?
No, they were just looking at dentate gyrus for that. Yep.
Yep.
And Alex, you have your hand up. Are you still typing a question? Okay, looks like no question. All right, 2E to G. These provided direct visual evidence of new hippocampal ensemble recruitment during systems reconsolidation using a longitudinal in vivo imaging method. And here the authors performed chronic 2-photon imaging in FOS-GFP mice with 3 key points worth mentioning. They implanted a cranial window over CA1 that preserved intact hippocampal circuitry. This is something that would be very hard to do in a human, but it's more accessible there in the mice. They validated that FOS-GFP faithfully reports endogenous CFOS activity. That was again in their supplemental material. And they established an imaging protocol that tracked the same neurons across multiple memory events. So then they carried out this longitudinal tracking that's shown in figure 2G. So using the system, they visualized granule cells, that's in the dentate gyrus, across initial training, remote recall, 21 days later, the post-recall test, and novel context exposure. And the critical finding there is that they were able to observe at single cell resolution how different neural ensembles were recruited across those memory events, providing the first direct evidence that systems reconsolidation involves new ensemble formation rather than simple reactivation of original engrams. And this in vivo imaging approach complements the FOS tree labeling that we saw just before by showing dynamically how remote memory recall engages a distinct hippocampal ensemble for subsequent retrieval. So here, figures 2H to 2L provide quantitative evidence that remote memory recall recruits new hippocampal ensembles. This is with their single cell tracking approach. And as mentioned, they were tracking neurons, the activation history of neurons during training, remote recall, and post-recall test. And the key findings here are that training activated neurons were rarely reactivated during remote recall or testing. However, neurons activated during remote recall were significantly reactivated during testing. And the reactivation rates matched previous reports for recent memory. So the data clearly show that remote recall recruits a new ensemble in dentate gyrus, that the new ensemble is preferentially reactivated for subsequent tests. The original training ensemble remains inactive. So these findings, they demonstrate that systems reconsolidation involves active ensemble replacement rather than simple reactivation of original memory traces. Moving on to figure 3. Figures 3A to D demonstrate the functional necessity of newly recruited hippocampal ensembles in remote memory retrieval. And the author has developed a dual optogenetic approach here, using FOSS-TRAP to label and inhibit training recruited ensembles, and FOSS-TET-TAG to label and inhibit recall recruited ensembles, so training versus recall. The key functional tests are selective inhibition of original training ensembles, and they showed that that had minimal effect on remote memory retrieval. The inhibition of recall recruited new ensembles, which significantly impaired memory performance, and the parallel inhibition of both ensembles, showing a dominant effect of the new ensemble disruption. And the takeaway message from that is that these loss of function experiments prove that the newly formed recall recruited ensemble is functionally essential, that the original training ensembles become dispensable for remote retrieval, and that systems reconsolidation creates a behaviorally relevant new memory trace. So that was the loss of function version. And here we have the data in ENF for the functional role of new versus original ensembles. So the findings are inhibition of recall recruited ensembles, impaired freezing. It's figure 3F, sorry, starting on the right. Confirming their necessity for retrieval, and inhibition of original training ensembles had no effect. That's the one on the left, figure 3E, showing they became dispensable. And yeah, so for broader consistency, the trap only inhibition replicated those results, that they did that in their supplemental material. And the experiments demonstrate that remote memory retrieval depends on newly recruited hippocampal ensembles, not the original trace. And the findings are robust across methods, so optogenetics and synaptic silencing, and are specific to remote memory. In figures 3G to K, sorry, good time for a question. Yes, Ken?
Sorry, I might have got lost, but I want to ask one particular thing. So,
Yeah.
It sounded like what you were saying is that the hippocampus is needed for remote memory retrieval, and I thought that that wasn't the case. That if you destroy the hippocampus, a different experiment, if you destroy the hippocampus, then you're still going to have the medial prefrontal cortex.
Sorry, this is where terminology gets difficult, and I even have problems with their terminology about remote memory, which I would consider the consolidated trace. When they're saying that the memory retrieval depends on newly recruited hippocampal ensembles, not the original trace, they mean that retrieving that memory into hippocampus depends on the newly recruited ensembles. So,
Okay.
So this is purely about retrieving into hippocampus, not about the ability to retrieve it at all and use it.
Okay, and there's always this there's this remote retrieval step that is occurring. So it's kind of like you've trained it. It was momentarily in the hippocampus, but actually that is never important in these studies. So you train it. It gets transferred from the hippocampus to the frontal cortex. It's living in the frontal cortex. Then you retrieve it from the frontal cortex 21 days later or 14 days later, depending upon the study. And then it becomes, again, dependent upon the hippocampus.
It becomes dependent on the hippocampus for the sorts of things they're testing, like the context specificity and the updating, these types of things. It doesn't mean you can't use the memory without the hippocampus. So I like to think of this as, as if you have a system. Let's think of a computer, and you've got a big memory system there, and you've got a cache, and that cache is active and is necessary when you're establishing new memory. But this cache is also there and is active and is doing stuff all the time, like when you're in a certain mode, right? Like when you've got theta. And then even when you're just retrieving memories that you've already stored that are consolidated, well, sure, the cache also tries to fill that. And then if the cache is there and you're putting something in that cache, then you can do some cool things with it. Because this cache, the hippocampal cache, it's not where you store all the details of the memory. You can't store all the details of vision and auditory and all those other multimodal things in hippocampus specifically. It's like a cache of pointers to all that other stuff. But you can set up some new pointers in there and use those to set up different relationships between things. So that's how I think about it. It's like this availability of that cache that you can do cool things with. And when they're talking about retrieval, they're talking about the retrieval into that cache.
And I just want to make sure on this particular figure 3, they are showing that they can inhibit the hippocampal engram, and this is the question, and then the mouse, the rat, reverts back to the generalized remote memory in the frontal cortex.
Yeah.
Okay, so this is another way of thinking of it is this shows that frontal cortex memory is still there, and it's just been being overridden if there's a cache hit, on the hippocampus side. Okay.
Yeah. I'm sorry that I did that I brought up cache, because now we're going to overgeneralize this metaphor.
I love the metaphor of a cache hit. I don't know if anybody else does, but I love that.
All right. Did we already do this one? No, we didn't do this one yet, right? G to K. So figures 3, G to K, this is now the opposite of the previous one. We had the loss of function studies. This is now gain of function evidence for new engram functionality. So here the experimental design that's shown in G and I. This is just giving you, the visual, but G is the actual experimental design. They used TRAP to label neurons activated during remote memory recall, the engram group, and a neutral context exposure control group. And they expressed HCHR2, an optogenetic activator, in those ensembles to test their memory encoding capacity. And then in J and K, we're getting the key results. Artificial activation of recall recruited ensembles induced freezing in a novel context, confirming they encode fear memory. And the neutral context ensembles, the control, evoked no freezing, proving specificity. So the takeaway is new hippocampal engrams formed during reconsolidation are both necessary, that's figures 3E and F that we saw before, and sufficient that we have here in the gain of function thing, J and K, for remote memory retrieval. And it demonstrates a functional shift from original to new ensembles. So these gain of function experiments causally prove that new engrams encode memory content, and they complete the story by putting that together with the loss of function work as what they're calling conclusive evidence. So then we move on to figure 4. This is about memory updating through hippocampal engram reconstruction. The behavioral paradigm in A here. So mice were conditioned in an original context, OCTX, and later exposed to a modified context, UCTX, during remote recall. The key manipulations that they carry out, so in recall in UCTX, the updating group, versus OCTX for the control group, and testing 24 hours later in both contexts. So they counterbalance that for the order in which they did the first part of that test, test 1 and test 2. The key results shown in figures 4B and 4C. So there's memory updating going on. Mice doing recalling in UCTX showed higher freezing in UCTX versus OCTX during testing. So they have new context preference. and it demonstrates the incorporation of new contextual features into the original memory. And then for the specificity of updating, they did some extra work in supplemental materials not shown here. They showed that no updating occurred if recall happened in a neutral context, and that updating required remote memory reactivation and not just exposure. The mechanistic insight is that updated memory retained contextual specificity, confirming reconsolidation reinstantiates hippocampal-dependent precision, new engrams integrate past and current information. And this matters because it's direct evidence that systems reconsolidation rebuilds hippocampal engrams, not just reactivates, that it enables memory updating with new information. And if you want to put that in a clinical context, it could explain how memories become malleable post-recall. And it could also explain something about false memory, because this mirrors the false memory mechanism. Then in figures 4D to I, mechanisms of memory updating, how does the hippocampus enable memory updating during reconsolidation? And what they show is the role of hippocampal activity in figures 4D to F, this one here. So they had the paradigm. They inhibited dentate gyrus during remote recall in UCTX in that modified context. That's 4D. And the result was blocking dentate gyrus activity. We're not talking about protein synthesis here. Prevented memory updating. And the specificity, well, there was impaired discrimination between OCTX and UCTX when this blocking was going on. And then in figures 4G to I, this is where they're doing the protein synthesis test. The paradigm is blocked protein synthesis in dentate gyrus during recall. The result is updating failed. So in H and I, that mirrors what we saw with the activity blockade. And the takeaway is that hippocampal engagement is mandatory. Both neural activity and protein synthesis in dentate gyrus are required to integrate new contextual details. And that's a mechanistic symmetry. So updating shares mechanisms with the reconsolidation that they showed in figures 1 to 3, confirming a unified process. So figures 4J to L, which is about the neural basis of memory updating during reconsolidation. The experimental approach here is the authors used the FOSS trap system to specifically label hippocampal ensembles activated during remote recall in the updating context, that's UCTX. And the key findings are memory retrieval. For the memory retrieval requirement, that inhibition of recall recruited ensembles impaired standard memory retrieval in figure 4K here. And for updating specificity in L, that these same ensembles were essential for updated memory expression. And this brings us to a unified model that remote recall in a modified context reactivates the original memory trace, recruits new hippocampal engrams. That these new engrams incorporate updated contextual information and become critical for subsequent retrieval. And I mean, that's a bit similar to how it's critical for the generation of brand new memory. So really, the hippocampus is just doing what the hippocampus does. And it creates an integrated memory representation that preserves both old and new information. And this critically, so what they think is the most important message here, is provides direct neural evidence for memory updating. It explains how memories maintain accuracy while incorporating new experiences, and it supports reconstruction-based theories of memory. There's a bit more about that accuracy maintenance that's coming up, because they haven't yet talked about neurogenesis, which is what we're getting into next. So here, figure 5, and just to let everyone know, there are 7 figures, so we're now past the halfway mark. In figure 5, A to E, adult neurogenesis enables memory updating through engram replacement. The experimental design here is to use X-ray irradiation to selectively block adult hippocampal neurogenesis. And they tested treatments on either 10 days pre-training or 1 day post-training. And they confirmed in a separate study that's not included right here that this indeed completely ablated newborn neurons when they did the X-ray ablation. The key finding is the critical role in memory updating. So X-ray treated mice failed to show normal degeneralization after remote recall. That's, oops, on the next slide here. And there was also impaired contextual specificity, shown here in H. The specificity of effect, well, there was no impact on baseline remote memory recall. They did that in a separate study, and it preserved recent memory generalization. The mechanistic insight here is neurogenesis mediates two key processes, both silencing of original engrams, enabling forgetting, and integration of new neurons into updated engrams. Oops, sorry about that. The model implications are that it explains how new engrams form during reconsolidation, and it provides a developmental mechanism for engram turnover. Let's see, Ken? Yep.
Sorry.
Don't apologize. It's fine.
Okay, the original training, so the X-ray ablation that caused no new neuronal genesis in the hippocampus, did that, I'm sure it's right in front of me, but I can't understand it. Did that affect the original training? Because the hippocampus would have to be working for the original training to even create a remote memory in the frontal cortex to begin with, right?
So I'm going to throw in something that is my own personal interpretation rather than something I know from the paper. You can't rely on neurogenesis as the source of, if you instantaneously, like in a moment, if you have activation of some patterns and then you're trying to create a connection with something that is happening in neocortex, you must rely on existing connections. So on something that just happens to be there, like coincidentally co-activated cells in hippocampus and in neocortex, and then strengthening those to set up your initial pattern if you're trying to capture something in the moment, a single presentation during an event. So this neurogenesis, bringing in these additional cells that have to then set up connections, that's very useful if you want to distinguish, if you want to kind of make fresh cells available that haven't yet got strong connections that are already involved in prior learning. So those are useful in that sense as a way to set up this new learning that we're talking about right now. But it's not clear to me that you would need them or could even use them unless they had already reached the maturation level where they had established new connections, so that they happen to have connections already that could be strengthened when you're just in a moment showing something, because the time taken for these things, it's just, you can't wait for them to establish connections, these new cells. So they have to be a bit older.
So you don't find that it's weird that it doesn't affect the original training, which I also wouldn't think that was weird. But
no, I don't think that's weird. No,
but apparently it does affect the I don't know what it's called. Okay,
this reconsolidation.
Reconsolidation. Yeah. So it does affect.
So and that may sound weird. Like, why does it matter there? And this is just me hypothesizing. So way back when we were doing modeling of these systems, and I was wondering, how does that neurogenesis in dentate gyrus fit in? What does it use? When we were doing testing of models where we had overlapping patterns of activity, which you would have if you're reactivating a similar context or you're reactivating a known memory. If you're overlapping a lot and then you're not doing interlace training, then new memory can blot out old memory. But what you really want is you want to be able to distinguish and learn separate contextually specific relations to something you've learned. And if you're trying to do that just from sort of this is not really neuroscience, this is just neural networks. If you want to do that well, then you want to try to separate those patterns as much as possible. So if you have fresh cells available that can now get introduced to this reconsolidation process that weren't already involved with the initial learning, I would assume that can be useful. So I just, back then, I hypothesized that the neurogenesis there was something that would make it possible to have crisp, more sparse pointers in the cache, if you think about it that way. I don't know if that aligns really well with what they're trying to, with the story they're trying to tell here, but that's how I try to think about why is it useful to have neurogenesis there? Because you've got these previously uninvolved cells. They give you something crisp and clear, not previously connected with other patterns. You're not overriding anything if you're not doing interlaced learning. But again, this comes from just neural networks and not from their study.
But just one thing relevant to that is when you have memory events and there's fresh neurons available in the, at least in mouse hippocampus, they tend to get preferentially recruited into new ensembles. So like every time you're forming a new memory in this sort of context, it's probably, if they're there, they're like new neurons that are being used for that. Which
That's
I
a
guess
very
is
valuable
in keeping
point.
with what Randall's
Yeah.
saying.
Because that fits nicely, because then if you don't have these fresh ones available, that's going to make it harder to set up this new context learning and reconsolidation. But if you do happen to have them there, if they've already matured enough and now you can use them, then that'll give you your extra.
You got to shift it. They're like NMDA receptor properties and stuff, so that you get a change in their excitability over time. Not that they're needed, as you can see in G. Like you can destroy them and the animal still learns.
Right. But so that, I mean, this all is cool, and I like that kind of thinking. But of course, this is us speculating and not directly related to what they were studying here. So there's a part here that they didn't look into.
Although in G, they're showing, I'm interpreting that as relying on the frontal cortex memory.
No, I mean, sorry. Relying on it, meaning
because
you don't get the new updated context specificity.
It's still
right?
general.
Yeah. Of course, that makes sense. Yeah, because for those existing memories, you would no longer need the hippocampus. It's just for the updated, like you're doing work with the memory. And once you're doing work with the memory, you need the hippocampus to make new versions of what you want to store, new information in there. But the old stuff, it's available through prefrontal cortex. Yeah, that makes sense. Okay, where were we? Have we gotten through this one yet? I'm not sure we had. Oh, no, we hadn't yet. Okay, so this is a bit about the age-specific role of newborn neurons in memory updating. They use this precision targeting approach of theirs, or another precision targeting approach using this NES-CRE R26 on double transgenic mice to selectively inhibit either 4 to 6 week old newborn neurons, immature, or 8 to 10 week old adult born neurons, mature. So we can imagine what those neurons, how they might differ. There are 2 inhibition time points during remote recall affecting reconsolidation and during post-recall test affecting retrieval. The key findings here were during the critical developmental window, so inhibiting 4 to 6 week old neurons during recall prevented degeneralization, so prevented the specificity learning, blocked that contextual specificity restoration. For 8 to 10 week old neurons, it had no effect. So in the retrieval phase, they had another requirement, newborn neuron inhibition during the testing also caused generalized memory expression, that's in M here, and loss of updated contextual precision. So the mechanistic insight there is immature neurons, 4 to 6 weeks old, uniquely integrate into new engrams during reconsolidation, just as Ariel already pointed out, and support retrieval of updated memories. And mature neurons lose that plasticity, they say. Then figures 5N to Q, neurogenesis controls engram turnover. The experimental setup here is in N. So again, we're using X-ray irradiation, and they used TRAP2 mice to permanently label original engrams, so training activated dentate gyrus neurons, and they tracked their reactivation after systems reconsolidation. They used the X-ray irradiation that blocked adult neurogenesis before memory updating. And then they tested memory performance in the original context and in the updated context. Their key findings are that in the sham mice with intact neurogenesis, that shows successful memory updating, a preferred UTX context. And in the X-ray mice, no neurogenesis in there. It failed to update memory. There's no updated context preference. The original engrams remain dominant. So neurogenesis ablation, so getting rid of neurogenesis, in a sense, unlocks the original engrams. With the neurogenesis intact, the original engrams were silenced, rarely reactivated. And when neurogenesis is not intact, after X-ray, the original engrams were reactivated during retrieval. And the interpretation there is that the new neurons somehow suppress old traces to enable new learning. How that works exactly, so what the mechanism is, I don't know. Okay, so figures 5R to U. These are, so it's updating requires neurogenesis. The sham mice showed that updated memory in UCTX happens, so new engrams dominate. And in the X-ray mice, it failed to update, stuck in the original engram. And the unified model is then that neurogenesis mediates some kind of a switch that silences outdated engrams, which I still don't quite understand, and recruits new neurons for updated memories. So you get engram turnover and contextual updating. And then they connect that in further implications with something like, okay, this might be how the hippocampus balances memory stability versus flexibility, and maybe linking theories of neurogenesis with both forgetting, clearing old traces, and learning, encoding new information. But I think there are clearly still some mechanisms, some details of that that are missing for me anyways. So then in figure 6, as we approach the end of the paper, they're getting into this question of how the medial prefrontal cortex to hippocampus circuit mediates reconsolidation. And we see here in A and B what they're doing. So they've got some retrograde tracing that they're doing in figure 6A. They injected Cav2 CRE into dentate gyrus and the viral vector into medial prefrontal cortex. And this then revealed the medial prefrontal cortex to dentate gyrus projecting neurons. And then they have anterograde tracing, where they're injecting the AAV CRE into medial prefrontal cortex and the other viral vector into dentate gyrus to confirm axonal projections to dentate gyrus. And the key implications here are that the prefrontal to dentate gyrus projections structurally exist, first of all, and that they functionally drive dentate gyrus activity. And that then they could be used, which is coming up in the next part, as a likely pathway for the integration of new contexts with valence. And that's where prefrontal cortex is going to play a critical role during updating. In 6G to J, they're investigating how the circuit from prefrontal cortex to dentate gyrus drives memory updating. They have 2 inhibition approaches. There's direct axonal silencing. That's where they're using the viral vector in prefrontal cortex to inhibit terminals in dentate gyrus, so these terminals here. And cell body silencing, where they're using Cav2 CRE and dentate gyrus and a viral vector in prefrontal cortex so that they will inhibit only the dentate gyrus projecting PFC neurons. And the key result here is that for both manipulations during remote recall, they blocked degeneralization, impaired contextual specificity. And the interpretation of that is that the prefrontal cortex to dentate gyrus activity is required to update memories with new context. And that has some functional insights that while the connections from prefrontal cortex to dentate gyrus are relatively sparse, They are probably critical in the sense that they probably have a modulatory, not direct effect on encoding. And the proposed mechanism they have for that is that prefrontal cortex input tunes hippocampal activity to enable new engram formation and the integration of context and valence. They didn't get into the details of that, and I imagine this is where things like mode switching, different things that happen in the presence of acetylcholine and theta is all going to play a role in one way or another, because it has to connect back to that evidence as well. But that's not something they looked at. Then in figure 6K to T, they're looking at the hippocampus to amygdala coordination, or hippocampus and amygdala coordination, and how that enables memory updating together. They use these inhibition strategies. So they've got two different ones. One is simultaneous inhibition, where they're allowing coordination so that coordinated activity is allowed between amygdala and dentate gyrus, and alternate inhibition, which prevents the coordination. And what they did is they targeted a bunch of different circuits. We're looking here at 6L, where they're doing inhibition of the direct connections from amygdala. So direct inhibition of amygdala and of dentate gyrus. We've got their manipulation in 6N, which is affecting the connections from prefrontal cortex to amygdala and the connections from prefrontal cortex to dentate gyrus. Then in 6P, they did a manipulation where they're affecting the PFC engram cells and dentate gyrus. And then in R, the PFC to amygdala projections and the dentate gyrus. And just summarizing this, the key findings are that amygdala and dentate gyrus coordination is essential. So if they use the alternating protocol, so that no coordination is possible, then memory remained generalized. It failed to degeneralize. When they use the simultaneous protocol, then they can get normal degeneralization. And the prefrontal cortex appears to orchestrate this coordination. So disrupting the PFC connections to amygdala and dentate gyrus, that blocked reconsolidation. So then their unified model here is that remote recall triggers PFC-mediated synchronization of new hippocampal engrams, the context, and amygdala valence engrams, which you might call emotion if you want, but I think valence is a better word. So this hippocampal amygdala coupling enables memory updating with valence. And then finally, we reach figure 7, the reconstruction model of systems reconsolidation. So it's I'll start with a core findings recap. Engram turnover. Remote memory recall recruits new hippocampal engrams. That was in figures 2 to 3, not original traces. It's enabled by adult neurogenesis, silencing old engrams. That was in figure 5. Then circuit mechanisms. The PFC orchestrates synchronization between new hippocampal engrams, the context, and amygdala valence engrams. That was in figure 6. And memory updating, new information is integrated via reconstructed engrams. That was in figure 4. And the reconstruction model that you're seeing here is then that before recall, remote memory is neocortex dependent, kind of GIST-like. I don't know why they're calling it GIST-like. After recall, hippocampus re-engages. It recruits new engrams for current context. So you combine the new current context with what you already know. The PFC amygdala dentate gyrus coordination binds a new context with valence. And then the outcome is accurate updating, degeneralization with correct information, or false memory if misleading information is provided. And so then on a broader scale, the implications are this may resolve long-standing debates by reconciling system reconsolidation neocortical storage with hippocampal involvement in remote recall. And it supports multiple traces or scenes reconstruction theories. It explains cognitive phenomena like memory updating, adaptive integration of new experiences, and false memories, misinformation being incorporated during reconsolidation. And then clinically, they suggest that that might have a meaning for interventions for PTSD, which is then maladaptive updating or memory disorders. And I don't have a specific slide here, but I do want to say something as a summary and also as a bit of an evaluation of this in terms of what does this mean for the memory decoding challenge. So first of all, summary. This work bridges three key themes. Dynamic hippocampus neocortex interplay, that remote memories aren't static, they're reconstructed upon recall. That neurogenesis is used as a reset mechanism, that it enables cognitive flexibility by silencing outdated engrams, which is still slightly mysterious to me. And then the potential involvement in false memory vulnerability. The price of that memory malleability is a distortion risk. And that shifts the paradigm from reactivation to active reconstruction. It's an interesting detailing that they did of potential evidence and mechanisms for silencing of original engrams as the new ones are created. It's not exactly direct evidence, not showing exactly what that mechanism of silencing is, but it's interesting anyway. The whole process here fits into a model of generating crisp, low interference engrams with context and valence, and the re-engagement of hippocampus as a way to make consolidated memory malleable, updatable, which makes sense given that the hippocampal system and its mechanisms are already available for those purposes during initial learning. And yeah, as we mentioned, these initial engrams have to come from existing paths, so mature cells through coincidental coactivation, but generating fresh engrams can prevent overriding without interlaced training and strengthen the availability of new memories in remote neocortical locations. Now, from the perspective of the memory decoding challenge, I'd say this work definitely fits into the detailed exploration of encoding and decoding mechanisms, given that the focus here is on hippocampus, which is not the location for detailed granular engrams of multimodal information, but rather it's a region that's involved with the manipulation of neocortical memory. It's not a study that reveals the identification or coding of those multimodal neocortical memory engrams themselves. Nevertheless, I think it was a well-conceived and valuable mechanistic study of mammalian memory systems and neural coding. And I am very interested in the decoding and encoding mechanisms. So for me, this is quite interesting, though with some questions open. Thanks, everybody. That was the extent of all the figures, and I'm sure there are probably some questions. Or at least some expressions of support. Thank you.
Maybe one question. I'm not sure if you're the right person to ask, Randal. Does a paper like this have any bearing on whether there's anything stored in the hippocampus long term in terms of storing remote memory? Just in terms of, I feel like I've seen studies that have gone back and forth on the degree to which the hippocampus actually stores long-term memory versus is just required for recall or for reconsolidation or any of those sorts of things. And yeah, maybe I'm just I'm still a bit muddled on that after having done this particular.
I didn't see anything in this paper that directly addresses how long cells used in the initial learning are still available for those same meaningful memories as a recall mechanism. So I don't think it directly addressed that. Although they did have the bit where they said it didn't reactivate the initial training cells. So whatever happened, what they didn't study in those intervening 21 days did affect whether or not those initial training cells in the hippocampus and the dentate gyrus, whether they were still going to react to this context that they were involved in during original learning. But it doesn't study what happened there, like that turnover, that initial bit, like why those initial cells are not really participating in it anymore.
But Randal, you're saying, I just want to make sure that I heard this right. When they got rid of adult neurogenesis, it was the original engram cells 21 days previous that reactivated?
Well, let's see. Let's go back to that slide.
Because my first reading of this, I didn't catch that, and so your presentation, I was like, oh wow, I missed I missed that completely. Which
Let's see
doesn't
where
say
they
anything.
have the actual. So the cell turnover, that's just the cells there.
I don't remember that specific figure
Yeah,
being.
I mean, if I interpret, sorry, Ari, you go first. I'm just
No, you go, Randall.
the way I'm interpreting Q here, because they're taking all the cells that they still see that are from training and from test, and then dividing by training. It seems to me like this is showing that after X-ray, that there are still more of the original training cells involved in the recall. I could be wrong, but that's how I interpret this.
Okay, and I mean that's pretty cool, right? Because it's kind of saying that, as Ariel was saying, I mean that's saying that memory in the hippocampus is still there. It's just that there is a new neuron, new engram waiting to be to form a new memory in the normal case. That will
still leaves open the question of how that silences the original engram, right? Like if these new neurons
there's
become
competition between engram groups.
yeah, so it
then
could be homeostasis in a sense that eventually gets that happen. If you have the new engrams taking on a large role, and then the old ones, I don't know. But it's still like lacking a very detailed mechanism for me, like something where you can actually follow what's going on.
I do remember seeing studies where if you block neurogenesis, as they talk about in this paper, you have a reduction in forgetting and then like new memory impairment. So it would make sense that blocking neurogenesis sort of keeps the old engrams around for just considerably longer, as shown by other paradigms as well.
And there's a second part to that, which is even if the old engrams are still available in this experiment after 21 days, if new engrams are not formed, it doesn't mean that they're going to be available forever. They could still become reused, because this is where other papers have described experiments about the reuse, that cells previously involved in one pattern or one set of stimuli in one context become involved in something totally different a certain amount of time later. I can't remember if they then also showed that the original context was no longer something those cells were involved in. I think they did. So it's possible that even just through the natural turnover of the hippocampus being engaged in a whole bunch of episodic memory creation, that there's some turnover anyway, and that those cells would begin to forget how they were connected with the neocortically stored cells, and that maybe we're just not seeing this after 21 days. But that's still not clear to me. So that's like mystery number 2 for me.
I know we're over time, so, but they don't say anything about this new memory that is updated, what that would eventually result in consolidation in frontal cortex. Do you have a speculation? Would that use the same engram cells in the frontal cortex, or would that also be yet a new engram that has nothing to do with the original engram?
If I had to speculate, I would say it would reuse cells that represent certain things in neocortex, because those are more directly connected with, say, auditory, visual, etc. So there's a more direct connection with something external. So my feeling is it would reuse those, but that the connections between them, so the autoassociative, heteroassociative connections that have some episodic, meaningful something, that have an episode, an event that they generate like that they would have new ones. And this is where you then get the dense mapping. So that's not sparse, crisp neural encoding, but a denser encoding where you do have to have interlaced training to make that happen. But that then ties all back right to studies about sleep and the roles of slow wave sleep and REM sleep in the consolidation process. and the potential that the way that that activity is reactivated in the hippocampus, that events do reappear in exactly the order they were in, but that the types of events that are replayed in hippocampus may be replayed in a manner that gives you that interlaced learning. Again, very speculative. But so in that case, you could do embedding new information without destroying old information if you do interlaced learning. So I have some speculations there, but it's mostly just, theory, no evidence.
I'm trying to bring this back. Also, I realize we're 15 minutes over time, so I guess people should feel free to leave if they want to at this stage. But I'm trying to think about this in the context of the memory decoding prize. I feel like the main impression I get from it is if I wanted to somehow read memories out of static brain structure, the hippocampus is not going to be a great target for me to look at because it's just changing all the time. It's not holding memories for very long. It's just pointing to stuff elsewhere as well. And that, like, cortical targets like the PFC might be better locations for more static, longer-term memory, but even those can be messed with by this sort of updating. But that's just like vague speculation. I'm curious as to what anyone else has on like what the implication of this paper might have for the prize or attempts to decode memories.
Well, one thing that I always come back to when decoding a memory, and I'll talk about this in 2 weeks with the next paper. Is any of these areas, if you can identify the collection of neurons that is an engram, that gives you something to hold on to. It's kind of like, okay, whether it's in the hippocampus or not, it's like this group of 1,000 neurons, let's say, that's interconnected, is all representing one memory. That's got to be able to now trace out to the different regions, let's say of the amygdala, and say, okay, well, what was this memory of? Was this a memory? Was this was this an auditory? Was this a fear memory? And so one of the real questions I have is how hard is it to look into the connectivity graph of a region like in the hippocampus and pick out engrams. Just neuronal engrams from the neuronal ensembles, regardless of understanding what memory it actually is encoding.
But if the turnover is fairly rapid, like let's say your cache, your hippocampus only has room for X number of episodes in some domain, and now we're a year later, so there's been a lot of turnover, and maybe because the duration of consolidation might be short enough that you only need LTP to achieve it, so LTP in hippocampus to have memories there temporarily, achieve that, and then they get reused because you could easily wash out the effect of LTP and then reuse them in another context. So you might not get much from hippocampus except for generally the pathways that exist to neocortex, and then maybe, I don't know, what you can pick up about exactly which patterns are pointing to at a given time. I'm just thinking out loud. I'm worried that it might give you only a small snapshot of what's available in the neocortex that you could trace that way, and that it's not clear to me whether you could trace it using methods that rely on, say, electron microscopy. But yeah, good question. Any other questions before we finish? And let me just quickly ask again. So Ken, you're the next one up, right?
Yes.
Yes.
Okay.
That's in 2 weeks. Yeah. Fantastic.
I have sent one invite to the Google group, so hopefully everyone who's here has received an invite to that next event in 2 weeks. Otherwise, we will send out some more invites.
I got it.
It's good. Okay.
Thank you, Randall. This was really fantastic. It was very, very in-depth, the discussion that you went through
Yeah.
the paper at. Yeah.
Well, thanks, everyone. It was a pleasure. See you all at the next Journal Club. Bye-bye.
Yeah.
Bye, everyone.
Thanks, Randall. Bye.